Okay, so my idea here with this presentation is to talk about endophytes in plants and really their significance. So I'm gonna, if I do this right, I'm gonna jump back to significance when I talk about things. What is it? Okay, if I don't forget that, right? If I don't forget that to do that, but let's go ahead. So microbial endophytes, of course, for anyone who does not know, they're microbes. And endophyte is a, oops, it's jumping. An endophyte, internal phyte plant. It is a microbe that goes inside a plant and does not cause disease. So we're used to microbes that cause diseases, but of course endophytes don't. I know much of the audience knows this already. Okay, but I still need to talk about it, need to say it anyways. Okay, and of course, we have our different kinds of agriculture, right? Our different approaches, I should say, to agriculture. We have one is a high chemical approach, and that chemical approach, we can call it conventional agriculture, and it uses lots of chemicals, uh, chemical fertilizers, uh, uh, herbicides, uh, fungicides, whatever there are. It's based on putting inputs into the crop and 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 onto the crop and onto the soil, and you get products out, and it works. Okay, but there's problems. I mean, the products are not as nutrient dense as they are when you grow things biologically. And then the chemicals you put out affect the biology of the soil and the microbial communities in the soil, and they affect the microbes in the plant and how the plant functions, how the plant works, putting those chemicals there. And also they degrade environment and degrade human health. Okay, many of those chemicals. And then we have our biological approach. Okay, and this is biological agriculture. And this has been, this is the ancient way of doing agriculture. And this is what people used to do when they first started doing agriculture is the biological approach. And that is without chemistry. Instead, soil microbiology is built up, soils are taken care of, the populations are increased in soil, plants are treated in such a way that they maintain their microbes. They tend not to be. We don't have fertilizers. We don't have things that are degrading microbes on seeds, for example, and in soils. And the products, the produce that is produced, the plant products are richer in nutrient density. Okay, and that's the other thing. This basically is the biological agriculture is growing plants with microbiology, and it's good for the environment. It's not bad for the environment. It's not wasteful. We don't have waste. We don't have chemicals that are going uh, into the soils and basically wasting those chemicals and doing damage to the soils. So basically, this is what Mother Nature does, biological agriculture. And it's better. It's better. Oops, it's not going forward. Okay. All right. And of course, one is inefficient. Okay. The chemical agriculture is inefficient. You have like uh, it's 60 to 70% of the nutrients that are put onto a plant are lost to the environment. And it's only 30 to 40% efficient, right? Mostly it's lost putting fertilizers in. Uh, uh, with biological agriculture, it's different. Okay, we plants get their nutrients either from directly from the soil, absorption from the soils, or through endophytes, endophytic microbes that go into plants. And the microbes, the plants get nutrients from those microbes. Okay, endophytes. Okay, I've already defined it. They are non-pathogenic microbes. And this shows an endophyte in a fungal endophyte in a grass actually, and you can see the hypha here of the fungus, and you see the cells in the background. And this is stained blue with aniline blue so you can see it. But these are non-pathogenic microbes that go into plants. And they have lots of effects on plants, lots of positive effects on plants. This one, by the way, is a fungus that's in fescue grass, tall fescue. They have some fungi in there that are also non-pathogenic to the plant, to the plants. Okay. Bacteria are also bacteria now. This shows bacteria. This is tomato. This is a root, actually tomato root, some cells there. And you can see right these root cells, these individual cells here, you can see the bacteria that are inside. This is a bacterium called Micrococcus. So it makes these little clusters of cocci, of little spherical clusters. That's the bacterium inside the cells. So the plants in these bacterial endophytes, they actually go inside the cells of the plant. I mean, they could be between the cells as well. They're definitely between the cells, but many of them are inside the plant cells, and they so in a sense they integrate with the plant, becoming a part of the plant. Epiphytes essentially become a part of a plant. And a plant is really not just a plant. A plant is a host to a whole lot of endophytes and other microbes that grow on it. And most of them are not damaging, not pathogenic. Okay, so the plant meristem. Plants take up microbes from soils, and the plant meristem is really where they take them up at, where they absorb them from the meristems. And I'll show you that. Okay, so so if you just look, I mean, I have this thing, meristem bioreactor. This is the idea that there's interactions between microbes and plant cells inside these meristems. But if you look to the right, you can see a root tip. This is a grass root tip, probably a POA. And this is the meristem right here at the tip. And you can see how blue this is right now. This is blue color. That's because I have a stain. We put a stain there that will stain the bacteria. And so you can see the bacteria forming biofilms on this meristematic areas there. And see here. Now this is good. This really is telling. These microbes affect, they provide not only nutrients, and we'll talk about that a little bit to the plant, but they also affect development of the plants. Plants do not develop properly without having endophytes, without taking in these endophytes, without having these bacteria, particularly bacteria, inside the plant cells. And if you look to the right, you see a root tip. Again, it's a grass, Poa pratensis, so it's Kentucky bluegrass. And you can see the tip right around here, and you can see the root cap cells falling off. You see them falling off, and that's how they work. They fall off. But what you notice is there's no blue there. There's no biofilm there. Also notice there's no hairs or anything there. If you look to the left, you see this one over here. You can see the tip again. Now this is with the bacteria, right? You can see the blue there where the bacteria are accumulating, forming biofilms, and you can see what it did. Look at all the hairs. The the hairs develop with the bacteria, but they don't without the bacteria. Plants need endophytes that they get from the soil and they can get from the seed that goes into seed, right, to develop properly. And if they don't have those endophytes, they don't develop morphologically properly. They don't develop their hairs properly. They don't develop their cells. Their cells are smaller. Their tissues are not as developed. Their tissues is not as tough. They don't have as many nutrients in them. I mean, we see the hairs, that's what we see, but actually, there is a whole world of physiological changes that go on in plants when these bacteria are in their cells. So they really need this. Plants depend on these microbes for their development and for their healthy development. Without them, if they don't have these endophytes in their tissues, they become uh, sickly, more susceptible to disease, and so forth. And I'll go over I'll go over some of that, but it's really, really interesting how dependent they are on microbes. And this whole dependency on bacteria started very early on in development of plants. I mean, I would say, I've done some work with algae, and algae also carry bacterial endophytes in their tissues. And if you look at the very earliest plants, the that were on the earth, they're like the mosses, the simplest of the plants, right? You look at those mosses and the liverworts, and you see that they also are covered and filled with bacteria. This is a moss shoot tip, and leaves are forming here. You can see the little leaves here, and you see this kind of I don't know, a hazy kind of a line here. This is bacteria in here. This arrow is showing bacteria on the surface. This tissue, these meristematic tissues in the leaves are covered with bacteria. So even the leaves, even the meristems, the leaf meristems, right, cultivate bacteria in them. And if you look at the at that moss, you can see the moss leaf again, the little leaf, and you see the little dots all in these cells. These are the cells, and you see the little dots, little blue dots. Those are all the bacteria. Okay, these plants, these tissues become filled with bacteria. So plants are not really just plants. Plants are plants and they're microbes. They have microbes. And why why do plants cultivate uh, cultivate these microbes? Why do they have these microbes? Well, for one thing, they've always had them, right? We know the algae, they're in the algae, they're in the mosses, the earliest land plants, they've always had them. They could rely on microbes for doing whatever microbes do. Okay, and so microbes can produce hormones and they can interact with the plant, causing development, causing increases, and triggering various processes in plants, even going into the nuclei around the DNA of the plant and triggering genetics of the plant, affecting, for example, one of the things that microbes do is to trigger, um, actually trigger the plant to express more genes. And what happens is you have uh, in the genome of plants and eukaryotes, many organisms, you have methylation that turns genes off. And what microbes do is they release that methylation, and that turns genes on. And what happens then when those genes get turned on is you get more, say, more expression of antioxidants, more of everything, really, more amino acids, more, it's just a lot more. It's the nutrient density is what we're talking about, the nutrient density. That's part of that nutrient density increase. And of course, with the rhizophagy cycle, and I'll talk about that, the plants can um, absorb microbes, oxidize them, and get some nutrients out of them from the soil. And so plants can use the soil microbiology as a vehicle to carry nutrients to the plant, into the plant. And then they absorb them and hit them with superoxide, with reactive oxygen, and extract, essentially oxidize the nutrients out of them. And I'll talk about that. Okay, so there also is this process of nitrogen fixation. I'm not going to talk very much about that. It's not, we know it's really important in legumes. It's not so important in other plants, but it still happens. Nitrogen fixation still happens. It's been shown. And what happens is these microbes are absorbed at the meristem, for example, the root tip. And then behind that root tip, they go into little vesicles called nitrosomes. And they've been shown, a couple of different studies have shown um, that in those nitrosomes, those bacteria are expressing the NIF, the nitrogen fixation genes, right? They're expressing nitrogenase, and they're fixing nitrogen. Okay, how important? Of course, it's not super important. It's not like legumes that are doing, they have a nodule, they produce a nodule and they reduce oxygen there and they very efficiently extract nutrients over a long period of time. This is different. This is when they absorb the microbes, the diazotrophs, they can go into these little vesicles, they can get nutrients out of them, but eventually these stop. They're not producing, at least not in these meristematic areas. All the nutrients are in these growing areas. And so it takes nutrients to fix nitrogen. That's where the nutrients are. That's where it'll happen. Okay. Okay. But, I mean, this is just one example, right? I mean, these things can go all over plants. And we do have some plants that are not legumes that grow very well with low nitrogen conditions. And those, of course, they have their own mechanisms for doing it. And I won't go into that. So what are these microbes? I mean, you've already talked about it. They're soil microbes, right? They're soil microbes. Essentially, they always come from the soil. Some of them go on the seed, and so they can be replaced. So these are microbes that if you maintain healthy soils, then plants can get these healthy, good microbes continuously from those soils. So soil really is the key. Yes, seed is the key. You don't want to, I mean, that's important. You don't want to sterilize your seed, and then your seedlings will not do well. So they will not start off very well. Now they can reacquire microbiology though from the soil if you have good soil. That's the most important thing is the soil. Okay, this is just a germinating seed, and you can see actually just shows a root coming out, and you can see yellow here. That's bacteria that are coming out of that seed, coming off of that seed, and colonizing these roots and the shoots as they develop. So there, there are a lot of there's a consortium, a little community is correct, community of microbes on seeds that the plants carry, or in seeds. This is tomato. Okay, people think they're sterile, these seeds inside the fruit of the tomato, but it's not true. They actually already have microbes in them. There are already microbes there. They have microbes. If you cut these seeds open, look at them with a microscope, you can see those little biofilms in there around the embryo. So they're definitely there. Uh, so I guess the point of this is you don't want to sterilize seeds. Okay, now I've alluded to the nutritional process that has come to be called the rhizophagy cycle or rhizophagy process. And here's some articles that we've written, and there's some before, and I'll show you. I'll explain the whole process. There was another. We didn't originate the rhizophagy process. It came out of Australia, and I'll show you that. Show you here. This is the group here. Rhizophagy, why the rhizo root, phagy eating, right? This is where roots absorb those microbes into their cells. And these are the Australian investigators that that described this initially. And their article was titled. It was in PLOS One. It's open access, so you can go and get it. In fact, most of our articles in this process have been open access, so you could find these articles online for free for no cost. But this was turning. This is in PLOS One and back in 2010, Turning the Table, the title, Turning the Table, Plants Consume Microbes as a Source of Nutrients, as opposed to microbes consume plants as a source of nutrients, right? Turn the table. Uh, this is what they described. And after they did it, we started looking at it. And we found that bacteria are involved in the rhizophagy cycle. They absorb bacteria, lots of different bacteria. Why they absorb one versus another one, we have no idea. Some just don't respond at all, don't go in, but others do. Now, why? We don't know. Yeasts also are absorbed, and this actually shows, these are root hairs here, and you can see the bacteria in the root hair. With the yeast, you can see them here. You can see the root hair, and you see these tiny little spherical things. Those are the bacteria inside. I mean, the yeasts inside. The yeasts are inside. They're little tiny little things. And see when they come out, you can see they're going out there. They're brown there, going out, and they're little here. And then as they grow, as they, as you know, as time goes past, increase in size. And that's because plants are putting exudate out, exudates, sugars, and other kinds of nutrients, sometimes amino acids there and organic acids. They put those nutrients out to feed those microbes that they're putting out of their cells from the root tips back into the soil. And this just and you can see they're growing. Those yeasts are growing there because the plant is feeding them. The plant is taking care of their little, their little microbial babies that they're having. But also algae can be go in, and we've done experiments with algae. And this is a chlorella, and you can see the algal cell in the center. You see it's green, but you see around it these bacteria. These are bacteria. These algae have their endophytes too. And what happens is if a plant takes in one of these algae, then the algae don't seem to survive very long, but you can see what happens. The alga here is inside a root hair. And you can see it's losing the green color. The chlorophyll is going. That's oxidation of that. And what happens as it's being degraded, the bacteria jump out. Look at the bacteria. There's all this roughage around here. That's the bacteria coming out of it. It's like rats jumping off of a burning ship or something, out of a burning building or whatever. They're jumping on all those bacteria are going out. So the plant is getting those endophytes. The plant is taking those endophytes. Okay, so this is rhizophagy cycle. This is the kind of the whole process. So the root tip is secreting exudates, and that's an attractant into the soil. Attractant exudates sugars and organic acids and sometimes amino acids and other substances that the plant secretes to attract these microbes. So microbes come swimming, come growing toward the tip, and then they cultivate them right there and they absorb them. And then they begin to extract nutrients from them oxidatively using reactive oxygen, superoxide actually, and other forms, but superoxide predominantly and other forms of reactive oxygen, but uh, superoxide's the main one. Okay, and then what happens is they will accumulate, they will trigger the bacteria inside the cell, the plant cell, triggers root hair elongation. And when those root hairs elongate, the bacteria get ejected. So you can see these hairs up here. Look at the little dots here. Those are bacteria coming out of the tips, coming right out of those tips. And then they go out to the soil. And so what happens is when these hairs grow, they're ejecting these bacteria out of the tips back to the soil. So that completes the cycle, right? It goes in, and then the survivors, some of the bacteria will break down completely, but many of them, at least the ones that are just ideally have the right oxidative resistance, they will survive. And then they can be cycled through the soil. And what the plant is essentially doing is farming these microbes, right? It is selecting microbes that it can use for as endophytes and for carrying nutrients. And it is increasing them inside the cells and then putting them back out into the soil. Okay. It's essentially engineering the soil environment, right? Engineering that environment using these microbes by increasing the populations of microbes that work with them. And so that's what's going on. And this is just a diagram by Harriet Mella. She made a little cartoon here that shows this roots growing and the bacteria going in and absorbing, and then they're losing nutrients when they go in from the reactive oxygen that oxidizes them, and then they go out of the hairs. Okay, and that's Harriet Mella up there. She's a very smart lady in Switzerland. But here, and if you consider like a corn plant, this is an illustration of a corn plant. You can see all of these little branch roots that are coming. Those are all branch roots. They all have their own meristems. Rhizophagy cycle is happening at every little meristem tip. So it's not just a few roots, it's a lot of root tips where this is happening. And so you could imagine, even though each tip there's just a tiny bit of nutrients coming in, uh, there's going to be a lot because there's going to be a lot of these root tips in different kinds of plants. And just to focus in on that a little bit more. If you look at now look at this. This is a grass. Here's the tip. You see the bacteria accumulating around there. You can see all the kind of irregular halo around that tip. That's the bacteria there. That's where they're going in. And then they're coming out of the tips. You don't see them coming out, but here you do. This is a fluorescent stain to stain the bacteria. I mean, the bacteria when they come out, and you see the hair here, and you can see them coming right there out of the tips. Here you can see another one. You can see there's little dots in there. Those are the bacteria. Those are protoplasts, by the way, inside. Those are protoplasts. They have no cell walls inside, either for the yeasts or for the bacteria. When they're inside, they're just naked protoplasts, these little spherical things. And when they come out, here, you can see they're starting to reform they're being ejected out. They're coming out little by little here, but they're reforming their cell walls and they're growing. So they're little when they're inside. Okay, this just shows a branch root of a grass, and you can see the root tip, and you see the bacteria here forming a biofilm there. This is the bacteria the plant is cultivating. The plant is cultivating these microbes. The plant is doing this. All plants do this. Okay, this just shows kind of the process a little bit. This is a root tip here, stained purple with crystal violet to stain the bacterial cell walls. And you see here where it goes in, you can see there's kind of spherical things. You can see them, but behind here, you can see where they're losing their cell walls. All this irregular, that's cell walls coming off right there. The plant oxidizes the cell walls off the bacteria, and we think that's where a lot of the nutrients come from, is right off of the cell walls. Bacterial cell walls will, this shows it again. See how irregular that is, how dark? Those are all the cell walls of those bacteria that are coming off inside the root cells, inside the cells. So the plant is degrading these microbes inside. Well, the nutrients we think are carried, at least some of the nutrients carried on the cell walls. And bacterial cell walls, you imagine in the soil, the cell wall has little ports on it, little binding sites where metals and things can attach. And that's what happens. And this shows a bacterium, and you can see, in this case, I think it's silver on the wall, little silver particles. So that's what's happening. And so when the plant takes those cell walls, they get the nutrients that are on those cell walls. That's what we think, how we think that's working. And, it may be, like, I won't say it's a huge difference. It's 10 to 30% more when we provide plants with microbes versus more of these, particularly these divalent metals, more than when we let plants uh, grow with reduced microbiology. So, I mean, we don't know what would really happen if there was absolutely no microbes there. We think plants would not grow, or even the plants may, they're certainly very susceptible to disease if they and stress if they don't have any microbes, because microbiology protects plants from disease and stresses. And but this is a root cell, and you can see these bacteria inside. See how they're spherical? They're spherical inside. They have no cell walls. They're protoplasts. They actually have a term for this, L forms, that mostly you see that in medicine, but you see it in plants too. And this is the case where we're referring to these L forms in plants, but they're just protoplasts. That's all they are. They're wallless bacteria. This just shows it again. One of my graduate students, she tagged a bacterium with Pseudomonas in this case with a fluorescent stain called M. cherry. And then she's scanning it, and you can see these little red things inside those cells. Those are the bacteria that she put in there. She labeled it and then fed it to the plant, and it went in like that. So, and where are these microbes going? They're inside the cells. They go in here. We used to think they just go in this, just underneath the cell wall, but actually they go all in here. They even go in the nuclei. And I'll show you that. They even go in the nuclei and around the nuclei. I'll show you that. So the plant uses superoxide to get nutrients out, right? And this is This is superoxide is basically oxygen. The plant gets oxygen from the air, so you got to have oxygen in the soil, for the plant to do this. So you got to have soils that have aeration, right? And in the cell wall, in the cell membranes, actually, they have this enzyme called NOx or NADPH oxidase. NOx is we used it also with animals and humans and so forth. The NOx, but these NOx enzymes will take oxygen, turn it into superoxide, and the plant cell will use the superoxide to get the nutrients out. And this needs iron, by the way. You can see as a cofactor. So if a plant is deficient in iron, it can't do this either. If a soil is flooded, uh, and the plant can't get oxygen, it can't do rhizophagy cycle. Okay, so there are requirements for rhizophagy cycle to happen. This just shows bacterium with cell walls, like a bacillus, cell walls removed, and you see there are these protoplasts. One of the features of these protoplasts, by the way, is that they grow real fast, and that the plant, with these protoplasts inside these bacterial protoplasts inside the plant cells, the plant can replicate them very rapidly. You know, with the cell wall, it takes, average 20 minutes or so for a cell to divide, right? But these protoplasts divide much faster. They just bud, blip, 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 like that, break them apart, and then you have, I mean, this is a form that the plant can replicate. So it's easier for the plant to replicate than when they have this form. Okay. But this is important for development. And this is what happens. This is a grass root. This is a grass root without where the microbes are removed. You can see the tip here, and you can see there's no hairs there. But there are little initials, root hair initials. But without bacteria in those initials, they never elongate. Okay, here's what happens when you put the bacteria on it. You see right away. You see the hairs forming right away. Here too. Look at this. And you see the brown in there. The brown, this is this is a stain for hydrogen peroxide, for reactive oxygen. And wherever the bacteria are is where the brown is. And you can see all these brown dots in these hairs, these brown areas. This is all where the bacteria, see these little dots all over. Those are bacteria in there. So these plant cells just get filled with bacteria. I mean, if nothing else that I do in my career is I will emphasize and continue to emphasize how plants manipulate these microbes and absorb them and become essentially one with the soil microbiology, and it's in that sense, you know, plants plants and their soils are definitely connected. I mean, we know people who grow plants have always felt like that, but the microbiology is continuous between the soil and the plant. Okay, not all the same microbes, but some of the same microbes. Okay, and there you can see one of the hairs. Look how full it is. All these little brown dots. Those are all bacteria. This is pseudomonads in there. Known bacteria from experiments that we do, where the controls show no hairs and no bacteria in them, And how do we do that? We sterilize the seeds to remove the bacteria, and we grow them in situations where it's totally sterile, so that the only bacteria they get are the ones that we put on them. Okay. This is tomato. You can see the same thing. We use antibiotic to treat the seed, and you see when we use streptomycin, you see there's no hairs on that root. This is a seed and a seedling root coming out. Here's with no antibiotic. You can see the hairs forming right there. Okay, and just the this shows the hair development. I mean, this shows the root hair development, and you can see where you can see the bacteria inside now. And where the bacteria inside, these hairs get longer and longer. And you get more and more bacteria in them because the plant is replicating them in there. Here's with no bacteria, you see a root hair initial, and you see nothing in there. Okay. Okay. Why is it growth of hairs and growth of cells linked to to the presence of bacteria. We think it is what I said earlier, and that is that there is a interaction, a hormonal interaction between the microbes and the plants. And it's not an, it's an interaction that plants require, that plants actively do this because it's part of their developmental process. Healthy plant has bacteria in it, growing becomes healthier and healthier because those bacteria are there. You remove them and plants become sickly and susceptible to disease. Here, this just shows a root without microbes, and you see there's no hairs. Here's a root. This is a poa annua of the grass, and you can see with the microbes, and you see hairs forming all over it. Okay, and why? We know that these microbes are secreting ethylene. We know they can secrete nitric oxide, and we think they're secreting nitrate too. Because we can see when we use the reason we think this is because we have histochemical stains that we use, and it's experimental. It's true. It's an experimental process. We need more work to show these hormones, that these hormones are produced around these bacteria. But with the histochemistry, it suggests that these hormones, these growth hormones that work in plants, are produced around these microbes, produced by the microbes. And I mean, you can see this inside the root hair. These microbes producing their hormones cause elongation, and the hairs grow. Okay. Okay, here's a sedge. I've shown this image lots of times. A sedge grows in rocks and grows even without soil. Okay, but what it does have is microbes. And if you look at one of the hairs, you can see these brown areas. Those are the microbes in the hairs, in that hair. And this is a living hair where the hair is working, and you see it moves the microbes, these shadows going around, moving those microbes around and around and around. That's what the plant is doing. The plant is processing these bacteria. They're being processed, and they're being broken apart. They're being increased. They're being replicated, and the plant is producing more and more of those microbes that it has. And these hairs, because of that, these hairs keep getting longer and longer and longer and longer. And when there's no more microbes in the hairs, then the root hair stops growing. It doesn't grow. And we can do experiments where we put different microbes in. Some microbes will make long hairs, some microbes will make short hairs. Okay, and that may be a hormonal thing, or it may relate to how the bacteria are able to stay in there in the root hair rather than being ejected soon. I mean, we don't exactly know what is causing that, but we do see different bacteria will cause different lengths of root hairs. Okay, this shows what happens to the bacteria inside the hair. You can see they're replicating them. These clusters, this is a root hair. These clusters probably came from a single bacterium that was replicated. And so this shows how the plant is increasing those microbes inside the hairs. Okay, this shows the movement. You see it going around. You see them accumulating at the tip. As they accumulate at the tip, what happens is in mass, and this is based on our experiments, the model that we have for how this works, they secrete those hormones in in bulk, right, in mass, and that causes a growth spurt. And when that growth spurt happens, these bacteria at the tip get ejected out. And you can see here's a root here full of bacteria. Some are stained, some are not. They're all in here. All this white stuff is all bacteria, but you see them coming out the tip. When they come out the tip, they reform their cell walls. All this wad of stuff, and you can see some are elongate. Those are forming their walls, whereas they're spherical inside. They reform their shapes, and so forth. And this just shows another one. You can see bacteria inside. You can see some bacteria kind of in transition going out, and you see bacteria outside being ejected. This is, I think, from basil, little herb basil. the model of how this works is, actually, I'll show you this. Okay, you have the root cell, the root hair cell itself here, and you see that it's doing cyclosis, what we call cyclosis or cytoplasmic streaming, and it just moves around and around. That's what I showed you before with the tomato root, where it's going around and around like that. This is the bacteria on the side. I made them blue, and on the tip, when they get to the tip is soft, and what happens is they pop out of the cell at the tip, out of the membranes, out of the edge. They pop out and they accumulate there, and then they jumble around. You saw them jumbling around in that last image at the tip, and then that's when they produce their hormones, and then that causes this growth spurt. And when that cell inside grows, it ejects bacteria, these protoplasts, right, out of these little holes in the tip of the hair. And then they go back to the soil, reform their walls and go back. And then the ones that are left in, I made them blue, they start to cycle around, right? The plant starts to increase them again. And then they accumulate in time at the tip and then again cause a growth spurt. And so you have root hairs growing in spurts like this as the bacteria accumulate in them. Okay, this kind of shows this. This is a bacterium. This is a root hair with bacteria in it where the bacteria weren't ejected. We took this bacterium from another plant. It didn't quite work with this plant. This was a Bermuda grass. We took it from, I think it was a Japanese knotweed. And what happened is these bacteria did not eject properly. And so you see them here in little banding where they banded areas where they accumulated and then they didn't go out like they were supposed to. And so this grew again and then they accumulated and you see them there on the edge and then they ejected. Well, they didn't inject very well, but they grew again and didn't eject. And you see that growth, but you can see the growth spurts. It becomes banded because of those growth spurts. That's tomato. That shows the ejection of bacteria from the tips of, and if you look down at three, down here, you can see there's three little pores, looks like three little pores, and these bacteria being pushed out. You can see how they're being pushed out. The tip, it's like ejection, like a syringe, putting them out, putting them out, pumping them out. Okay, this is um, it is logical that this is a nutrient absorption mechanism, another way and to get nutrients. And um, we know that plants get nutrients dissolved in soil water. They just absorb them. Uh, we know that plants can affiliate with mycorrhizae and get their nutrients, some nutrients from mycorrhizae. And now we know that plants can oxidatively extract some nutrients from bacteria, microbes that they absorb. Okay, so the how much, I mean, the plants are getting nutrients from all of these ways, right? All methods. And rhizophagy cycle, rhizophagy probably delivers the nutrients that are more difficult to get through absorption. That's probably the role of well, one of the nutritional role of rhizophagy cycle is get the nutrients that are hard to get. Um, but there are other roles. Okay, plants have to have those microbes for development, so they've got to absorb them for their health. They need them. They have to absorb them, like our guts. If we don't have our gut microbes, we get really sick. Plants get really sick if they don't have their microbes as well. Okay, so let's see what kind of. We've done some experiments to show that rhizophagy or the microbiology in the root tip is important, and some of those are like these uh, isotopic nitrogen ex tracking experiments. And these basically what we've done in these experiments is, let's see, what do I have here? So we've put some protein with isotopic nitrogen in the media, and then we grow them with and without microbes. You can see the grass here, right? And this grass, this little seedling coming here, the seeds were sterilized to remove microbes, and these weren't. They were germinated and so you could see the difference between these two. This is grown in this medium is protein that contains isotopic nitrogen, nitrogen 15. And we use nitrogen 15 because we can track it, because uh, it's a low level typically. Nitrogen 15 is a low level typically uh, in plants and in the environment. So they can, where we add it in the protein, if they absorb it, we'll be able to tell that they're absorbing the nitrogen 15 because we can measure it in the seedling. Okay, so you can look at the seedlings here. This is with microbiology to the left, without microbiology, or with reduced microbiology to the right. You can see the shoots are larger over there with the microbiology. You can also see the roots are secreting more reactive oxygen. This is stained for reactive oxygen. So the red color or the brown color is the reactive oxygen. So there's more reactive oxygen there. That also means because these plants have microbes in them, they're using more reactive oxygen inside the plant, and so they're upregulating their oxidative stress genes, their reactive oxygen management genes, their antioxidants, and the stress genes, because all stress is oxidative stress. And so they're upregulating those enzymes in those systems. They're growing better, they're hardier, stress hardier, they develop better. Okay. And so what is this experiment? Okay, so here we have, what do we have here? We have, okay, this is results, for example, what we're seeing here, protein, bacteria. Okay, so this is when we have bacteria, we get this much nitrogen 15 absorbed into the seedlings up here. Nitrogen 15, right? Absorbed that much nitrogen, essentially nitrogen 15. And then with reduced bacteria, you see it's this much. See, they're still getting some, okay, but they get much more with those microbes present. Okay, and the other things that, we've looked at we've done experiments. One of my students did an experiment where she looked at cotton, and with a particular microbe, a particular bacillus, she then measured uh, the physiological processes that are happening in the plant. And these are the genes that are upregulated, and what she has, nitrate metabolic processes. So nitrogen is coming in to the plants. That's the suggestion with those microbes are there. Nitrate assimilation, okay, more nitrate, right? Taking in more nitrate. Again, I talked about microbes producing these, and also this also come from the soil, improving soil absorption and nitrogen compounds. There's other processes like this microtubule base movement. And also this microtubule cytoskeleton. That's the cyclosis that we're talking about. They're doing more of this cyclosis, right? Moving those microbes around, replicating them. So plants get busy when they have microbes, and without, they don't. I guess they're getting, maybe they get bored without the the microbes speed them up. They can do more with those microbiology. So that's the okay. And um, this is some of the nutrients we think that they carry are these divalent metals, and these are the ones the many of the minerals. These that we think this is oxidative extraction. Some of these are really important. The ones that I have marked in red are usually very high when plants have microbiology, and much lower when they don't in our experiments. And in some cases, like manganese, it's like a 700% difference in whether they have if they have microbiology, microbes, you have a lot of manganese, and if they don't, you get very little, very little. Okay, so. There are these other concepts that I want to talk about. I know we're running out of time, Matt, but there are these other concepts. Like one of them is habitat adaptive symbiosis, and this is relates again to plants uh, and the agency of plants, right? Their capability to take care of themselves if they have microbiology. And this was a habitat adaptive symbiosis was a concept uh, proposed by my friend of mine, Rusty Rodriguez, and Regina Redmond, friends of mine. And this is the idea that plants will, depending on their stress in their environment, they will associate with microbes that help alleviate that stress. And they did, in this work, they did some experiments where they grew plants, a grass on a cline, or they studied the plant on a cline, and they found out in the cline that up high, it was a low salt area, and down low was a high salt. And so the plants were growing all along that. And what they noticed was when they got close to the salt area or in the salt area, the microbes in the roots changed and they got some microbiology. They got a fungus actually that was growing that would grow with the plant and then give it this resistance to salt, this stress, which is an oxidative stressor, right? A salt stress. So plants can get the endophytes that they need in order to optimally grow if they're somewhere in their environment. So this is another argument for making good, healthy soils, right? And the final thing I think it's well, I think it's the final thing I want to talk about. That is that um, the chain that microbes in plants cause genetic changes, or popular term now is epigenetic changes, but there also may be some actual genetic changes, some use of triggering transposons and so forth that cause plants to be more variable when they have lots of endophytes versus when they don't have many endophytes, depending on how they're cultured. So basically, plants, and this has been shown that organically compared to conventional growth, the organic growth, you show more gene expression, more epigenetic, more epigenetics, gene expression more gene expression is going on. Okay, and I've been working with Walter Goldstein on the Mandamin Institute on corn. On he's trying to breed this nitrogen fixing corn, and you can see some of his varieties here. You can see how dark green they are compared to some commercial cultivars there. So this is a work in progress, and we're trying to understand it as much as he's actually trying to create some products, but he also is a very good scientist. He's trying to understand this process as well. This just shows some of the corn, and I'm not going to go into the details here. It gets pretty complicated, but I will show this. The bacteria go in the nuclei. This is a potato. And you can see the nucleus here in the middle. You can see these around it. Those are bacteria. They go in the nuclei. They go around the nuclei. Here's this actually is I think it's a hosta, right? And you can see the cell, this epidermal cells. You can see the nucleus. These are bacteria coming out of these nuclei. You see them, they're going out of the nuclei. Here too, you can see them going out of the nuclei. So this is a stage of development, and they're in those nuclei. Now, maybe when these cells get much older, they're not going to be there. They're going to have all left there. But early on in development, they're there. And so while they're there, they're turning on genes, right? They're affecting genetics, and we don't exactly know how. We have some hypotheses, but that's what they are. But you can see here these things coming out, these little dots, those little vesicles, those are full of bacteria. Those little dots in the nucleus, so you could see them here. Here's another one. Look at the bacteria. Here's the nucleus. They're actually coming out. Look at coming out in little streams going through the cytoplasm over to the edge. It's a very dynamic kind of an interaction. And it would be difficult. It would be crazy for us to say there's no effect of these microbes in these on the genetics of the plant. That would be crazy for us to say that. They're in there. You have to be, and this is a problem with some of our some of our science is a lot of it is black box and we can't see what's going on. And so we're just guessing, and you might say, well, it makes no sense that anything would happen. But if you see it, that anything would happen to the genes, right? But if you see it like this, then you say, gosh, something's probably happening, and that could explain much of our epigenetic change that we're seeing, that direct interaction between the microbiology and the plant genes. And, well, these microbes protect plants from disease. They change behavior in fungi. Okay, and this is an experiment with basil seedlings. And I guess it shows these seedlings. What we did was, okay, so the upper two have bacteria, they're natural bacteria on them. And to the left, this is just them growing alone. And then to the right, we put Fusarium oxysporum, I think it was, on them. And you see it has no effect, a very minimal effect. It's not affecting these seedlings. Okay. And if you look below now, this is where the microbes have been removed. And when we remove the microbes, you see these seedlings were diminished a little bit, but they're still growing, right? Okay. But then when we put Fusarium there, there's totally damped off. It killed these seedlings. They can't grow. The bacteria that were on these seedlings are critical for the health of the seedlings. They protect it from disease. And I'm not going to go into too much of this, but it's really cool. The bacteria go into the hyphae of the fungus and alter the fungus behavior. They make these fungi less virulent, less deadly, less pathogenic, and the fungi then behave more as saprophytes rather than pathogens causing disease. And it's not that way for every single pathogen, but a certain number of certain kinds of pathogens, it is that way. And so you get a lot of these pathogens these facultative plant pathogens that are impacted that turn to saprotrophy instead. Why it shifts them that way, we don't know. Guess is maybe it has to do with nutrients that the bacterium gives nutrients to the fungus. It's just not clear, not exactly clear what's going on. Okay, so the point is that the fungal behavior is altered by the bacteria. And I'm not gonna, we did some experiments about that. Oh, but this is pretty cool because here you can see the hyphae, and you can see over here the little fluorescent dots there, the little dots in the hyphae. Those are the bacteria inside the hyphae. Fungi also take in bacteria, and when they do, it alters their behavior. So healthy soil. So you could imagine if you're creating a very healthy soil, a lot of the pathogens that you have normally in the soil, they're going to be there, but they're not going to be pathogenic to your plant. And your plant is going to be resistant to whatever the pathogen is. So it's a dynamic, multiple. This goes back to the soil health thing. I used to I've been focused doing science on endophytes for a long time, and I kept hearing, people talking about soil health, and it's really important, really important. But now I understand really how important it is to have this diverse, healthy, microbial rich soil. To This just shows more of bacteria coming out of shows more. This is another fungus. This is zygomycete showing bacteria coming out of it. Look at these little wads of bacteria. And you see the hyphae are just loaded. These zygomycetes are full, 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 full mushrooms. You can look at mushrooms. You can see the hyphae in the mushrooms. So, okay. And so I guess to summarize, I can do that. So why do plants need endophytes, right? Why do they need endophytes? They need them for nutrients, for one thing. Why do they need rhizophagy? The same answer. They need it for nutrients. Also, because plants become stress tolerant. They upregulate their oxidative stress because they're interacting with microbes using reactive oxygen. So they upregulate those stress genes and they become resistant to stress. They have fungal pathogens. They go into the soil and also in the plant and they affect the fungi. They cause the fungi to be non-pathogenic or less pathogenic, to have reduced pathogenicity, right? Reduced virulence. And the other thing that they do is they will go into the nuclei of the plant, developing nuclei, and turn on genes that make that help the plants adapt more rapidly to whatever the conditions are. It's genetic affecting, epigenetics, making plants more adaptable, more rapid evolutionary change, more rapid adaptation to whatever is happening in the environment, you know. So, I mean, these are what they're doing for the plant, right? This is what they're doing for the plant. For people, for people, of course, we're putting in less money. If you want, still costs something to grow regeneratively, right? That costs, but you don't spend as much on chemicals, and your plants are better. And people, we eat those plants, and those plants, those crops, the more the more microbial rich your soil is, the more microbial rich your plant is, and the more microbes you can take into your body, into your gut, and the healthier we can be. That's the simple association there. And that's, teaming with bacteria. This is Jeff Lowenfels, who wrote a book and on rhizophagy cycle and endophytes. And this is the popular book that he did. And he goes around. He's more prolific than I am at talking about this now. He's doing great. I need to be working for Jeff, supporting Jeff really in his efforts to do what he's doing. That's kind of what I'm doing, really. And these are people who supported this work, who did this work, who contributed to this work. And these are graduate students and visiting scientists and postdocs and so forth. And that is it. That's it. It for the stop to share. I did longer. I'm sorry, it was longer than I was supposed to do, Matt.
It was so good. It was so good. I do have a couple questions.
Go ahead.
I had someone hear about my work and then they were like, "Oh, I love biofertilizers. Here, this is the latest biofertilizer my friend's company just came out with. They're so proud of it. It's and then I read the label, it's Aspergillus fumigatus."
Well, you got to make sure it works. But aspergillus is a fungus that we do find in plants. So we'll find them on
100%.
plants.
But my concern
Yeah.
when I hear that though is the end of the season. When it's drying down the aflatoxins, the mycotoxins, fumigatus is aspergillosis, right?
Well, it comes very rapidly from in fact, the big source of Aspergillus propagules is actually agricultural fields. Yeah. So it contributes, it would further contribute. I'm tell your friend that. I mean, maybe they have a way. Like, I don't think that would be good. Your friend made probably they're working
Yeah,
their
I
fingers
told him
to the
not to
bone.
use it. I told my friend not to use his friend's thing. I was like, don't do that. There's better, partnerships out there.
Aspergillosis is a problem. It contributes to that's contributing to the bad reputation of agriculture because typically people who are immunocompromised in some way have problems with aspergillosis. Typically, okay, you may have oh, now I understand why you said that. Yeah, I see where that's
So,
coming from. See where
so
that's coming
my
from.
family is in that small percentage of people that has those haplotypes. We have both. And so we have a difficult time cleansing mold, so we're sensitive to these things. And yeah, it's interesting
Mhm.
because we hear more and more about those things on the wind from these agricultural fields. And I feel like it's exactly what you said though, where you're missing a whole ecosystem of biology that from the roots to the leaves to the soil cycling that will make it because I mean every soil test has fusarium in it. Every soil test that I've seen that people are sharing with me, it's there, but it doesn't behave like it's there when you have that diversity in ecosystem. And maybe for those folks
Yeah,
transitioning, that's what they should aim for.
yeah. And we don't know what happens when these bacteria go into a fungus like Aspergillus, and they may turn off one thing it does is it reduces, when the bacteria go in, it reduces the reproduction of the fungus, reduces the number of spores it produces. And so when you have lots of bacteria, lots of a good mixed soil, right? Mixed community microbes in the soil, you would expect fewer mold spores forming from that soil. And you can see that readily when you try to sterilize soils or sterilize seeds and soils. Then you see mold growing all in it. It
Yeah.
makes a it actually makes a huge difference. That's a good point that you made. Yeah, it's true. Absolutely true. 100% correct.
I am so excited to share this talk with everyone because I know that we've been I've been learning from you from years, but for years, and in this presentation you brought up like things in a different way. It seems like I'm really excited to share because I felt like I learned so much. The first time I saw algae was in sugarcane sap that someone sent me a chunk of sugarcane, and we were looking at it, we're like, that's green. And I've got that epifluorescence, so I reach in, and it causes all chlorophyll to turn blood red, and I pull it, and it turns bright red, and everyone's like, "Oh!" And yeah, it was algae, and then we're like looking it up live in real time, and it's so funny because first thing that comes up is like, yes, it's an endophyte. Second thing that comes up is your talk for end soil algae. So I was like, once again. James isn't even in the room, but I'm learning from you. So thank you so much for all your work. I know you may feel like Jeff's doing a lot of work, but you are doing so much work, and the community knows you and appreciates you.
Well, thank you, Matt. I appreciate that. Thank you very much.